there's a cell membrane in a plant cell, there's a nucleus in the animal cell, and there's also a nucleus in the plant cell. The only difference in regards to the nucleus in both is that the nucleus of an animal cell is at the center, while the nucleus of a plant cell is at the periphery at the end. Okay. Now the reason why we have it at the periphery on the side is because we have a subvacuole, which is basically a solution that controls what enters inside the plant cell and what leaves, because this solution or the subvacuole stores sugar and salt. So when it stores sugar, it makes the cell either highly concentrated or lowly concentrated in relation to whichever solution the cell is placed in. And we're going to talk about that when we'll be talking about cell physiology. So then we have a chloroplast in a plant cell, but in an animal cell, we don't have a chloroplast. There's a similarity in both animal and plant cell in regards to the cytoplasm. So both the plant and an animal cell have a cytoplasm, but when you look at the outermost layer, the outermost wall of a plant cell, it is referred to as a cell wall, and then just next to it, we have a cell membrane. When you talk about an animal cell, as you can see, we do not have a cell wall in an animal cell, but we have a cell membrane. What this suggests about a plant cell is that a plant cell is more stronger compared to an animal cell, and animal cells easily burst. The reason why they easily burst is because they lack a cell wall. But a plant cell has a cell membrane and a cell wall, so this cell wall makes it very strong. So no matter how much no matter how much water gets inside the plant cell, it will not burst because of the cell wall. Okay, so we also went ahead and talked about a plant cell and an animal cell as seen under an electron microscope, and we said that the reason why we see a lot of cell organelles inside a cell, those small structures which are found inside a cell, they are referred to as cell organelles. So the reason why we see a lot of cell organelles is because an electron microscope has a very high magnification, and at the same time it has very high resolution. And we said magnification is the ability to enlarge, make something appear bigger. So when you make the animal cell appear bigger, you're able to see what is inside. So it's like zooming it. So when you zoom it, you see exactly what is on the inside. And now this is where we have all these cell organelles. So the Golgi bodies, we have the ribosomes, the cytoplasm, the lysosome, mitochondria, the vacuoles, the nucleopoles, the chromatin, smooth endoplasmic reticulums. And if you look at a plant cell also, as seen under an electron microscope, you're going to notice there are also some cell organelles which are similar to those that are in an animal cell. For example, we have the ribosomes, we have the Golgi bodies, we have the smooth endoplasmic reticulum, we have the lysosome, we have the rough endoplasmic reticulum. However, you will also notice there are some now which are clearly visible that are not present in an animal cell. Like for example, there is this chloroplast here. So a chloroplast is only present in a plant cell. The reason is because chloroplast has chlorophyll, which traps sunlight for photosynthesis. So if plant cells, because it's only plant cells that can carry photosynthesis, especially the green plants, therefore you will find that animals don't have chloroplast because it is going to be insignificant since they do not carry out photosynthesis. You'll also see the cell wall here, which is absent in an animal cell, but present in a plant cell. So we went ahead and talked about a couple of these cell organelles and their functions, like for example, the cell membrane. We say that it is used, it controls what enters in and out of the cell, since it has poles which are going to control the entry. How does a cell membrane control entry? The poles are of specific size, which does not change. So anything that is able to pass through the pole, since it is small, it will pass through and get inside the cell. Anything that is bigger than the pole will not pass through the pole. So therefore, in biology we say if it allows some materials to pass through and others are restrained from passing through, then it is semi-permeable or it is selectively permeable. Semi-permeable means it allows some materials to pass through while others do not pass through. So a cell membrane is semi-permeable because it allows some materials to pass through that are small enough to fit in the poles and others won't pass through. We went ahead and talked about the cytoplasm, where he said is a fluid medium where chemical reactions take place. We talked about the mitochondria, where we said that the mitochondria is the powerhouse of the cell. This is where reactions take place that will provide your body with energy. Since we require energy for almost everything that we do, that energy must be supplied to our bodies and to our body cells and organs and tissues, and it is formed inside the mitochondria. So therefore, if it is a muscle, since muscles are used for working, because they require a lot of energy, you will find that they have a lot of mitochondria. Okay, so mitochondria tulisema ni zikiwa mingi, ikiwa moja ni mitochondrion. So for example, the biceps, the muscles in your arm, those muscles will require a lot of energy so that they can move. Okay, now that process of movement is going to require energy, and therefore the body has packed a lot of mitochondria in every place where we have a muscle, so that we can have a lot of energy being synthesized there to allow movement. Similarly to your heart, the heart muscles, you know the heart is the largest muscle in the body. So the heart has a lot of mitochondria because it requires to keep on pumping every now and then without fatiguing, without getting tired. So therefore, it requires a lot of mitochondria. So if you took a heart muscle, it would have a lot of mitochondria. So we went ahead and talked about other cell organelles like the endoplasmic reticulums. We said the rough one is connected to a ribosome. The ribosome synthesizes proteins, and then the rough endoplasmic reticulum transports the proteins. We also went ahead and talked about the smooth endoplasmic reticulum. We talked about the lysosomes, where we said that they contain a lytic enzyme and they destroy worn-out cell organelles and cells. So if you want to go through those cell organelles that we talked about last time, you can rewind and go back to the previous lesson that we had. It is available in YouTube, so you can watch. So let's talk about chloroplast. So we're beginning the lesson now. We're continuing with the cell organelles and their functions. Now, this chloroplast that you're seeing here, this is a chloroplast. It's a cell organelle, chloroplast. And if you remember very well, in an animal cell, we did not see a chloroplast. Now, why didn't we see a chloroplast? The reason why animal cells don't have chloroplast is because they do not carry out photosynthesis. This cell organelle you're seeing here, chloroplast, the reason why it is present in a plant cell is because it carries out photosynthesis. How does it carry out photosynthesis, or what enables it to carry out photosynthesis? So if you look at this chloroplast here, there are two parts which I want us to focus in. I want us to focus on the stroma and also granum. Let me show you here. We have the granum and we have the stroma. So we have the stroma and we have the granum. If you look at the diagram, looks like they are layers of stacked structures. So hata umeona imeandika stack of thylakoids. Okay. Now granum is very special because it contains chlorophyll. It is not the entire chloroplast that contains chlorophyll. It is the granum. Okay. Now, what is the function of chlorophyll has being present in the granum? The function of chlorophyll is simply to trap sunlight. Okay. So during the day when there is sunlight, so this is the sun and it is shining, the sunlight will be trapped by the granum like that. And when it is trapped by the granum will carry out photosynthesis on behalf of the plant. However, the process of photosynthesis takes place in two stages, like we are going to see. So the first stage of photosynthesis, we are going to have sunlight being used, and it takes place in the granum. So kindly note that in the granum, photosynthesis stage that requires sunlight takes place. However, if you look at them, all of them are inside. So that's why we say chloroplast is used for photosynthesis. But when you zoom it out, when you use a highly powerful microscope, like an electron microscope, you'll be able to see the insides of the chloroplast, and you will see that despite the fact that it looks like it's the chloroplast that carries out photosynthesis, there are some internal parts of the chloroplast which, for example, the granum absorbs sunlight because it contains chlorophyll. And then we have the stroma. The stroma is the white part. On the inside, this whole white part there. So the white part, which is the stroma, carries out another stage of photosynthesis, which we call the dark stage. Okay, we're going to look at that when we'll be looking at nutrition in plants. So then we have the starch granule. It's very simple. Starch granule stores starch. Now, one thing you should note is carbohydrates are stored in both plants and animals in different forms. So plants store carbohydrates in form of starch, while animals store carbohydrates in form of glycogen. So sisi ukiangalia kwa mili yetu and you want to trace some carbohydrates, it is going to be stored as glycogen. But when you look at plants and you want to investigate or test the type of carbohydrate that is found inside the plant, you'll find that it is in form of starch. That's why we have starch granule, so that it can store starch. So what happens is when plants carry out photosynthesis, they form carbohydrates, and those carbohydrates will be stored and they will be converted and stored in form of starch. So one thing else that is important for you to note is that the inner membrane and the outer membrane. So a chloroplast has two membranes. So we have the inner one and we have the outer one. But I'm insisting, the two single most important parts of the chloroplast in relation to photosynthesis are the granum and the stroma, because the granum will trap sunlight because it has chloroplast, and then you have the stroma which is used in the process of photosynthesis. We'll talk about that later. Good. Something else to note here is granum and grana. Granum is in singular, grana is in plural. So we have granum, like the one that is displayed here, and we have grana. So we have G-R-A-N-A, grana. Zikiwa mingi zinaitwa grana, ikiwa moja inaitwa granum. So it's important that you note that. Very good. So we continue. So we have said that the chloroplast is found in plant cells only, and they provide site for photosynthesis. So we move on to the next cell organelle. So I want us to talk about the cell wall. So a cell wall is only found in animal cells, if you look at both cells. And the reason why we've said that the cell wall is hard, it's because it is made of cellulose. So cellulose is a type of carbohydrate which is stored in the cell wall. So it is combined with the cell wall such that it makes the cell wall to be very strong. That is why nimesema, no matter how much water gets inside a plant cell, the plant cell will never burst, because it is so strong, since it is strengthened by cellulose. Therefore it does not burst at all. So why do we need to have a cell wall being strong? These are the reasons. So functions of a cell wall are reasons why a plant cell has a cell wall. Number one, cell wall protects the cell against mechanical injury. Number two, it gives the cell a definite shape. So no matter what happens, no matter how much water the cell gets, no matter how much water the cell loses, it will remain to have the same shape. And this shape is given by the cell wall. Unlike in animal cells, where if it loses water, it shrinks so much, and if it gains a lot of water and it is full of water, it will burst. But for a cell wall in a plant cell, it makes the cell of a plant to be so rigid such that it does not burst at all. So we also have the other function of a cell wall, which is it provides mechanical support. So there are those plants which do not have a wood stem. Now let me talk about this. We have two types of plants in relation to support. What is support? Support is basically what allows a plant to stand and remain active in the environment. Like if you look at a tree has wood, okay? But if you look at something like coriander, if you look at something like a coriander, something other plants like most of the flowers that we grow at home, something like passion fruits, something like watermelon vines. So those plants do not have a woody stem, okay? Instead, they are able to remain active in the environment by use of water, okay? So the cells gain water, they become firm, and since they cannot burst, they then allow the plant to remain active. We call it the plant remains erect in the environment. That is why if you have a herbaceous plant or a herb, okay, like an oxalis, when it is very hot, you will find that that plant will start actually wilting. Wilting knee, or the leaves will droop. This is a condition where you'll find the plants, since the cells have lost water, they start loosening up in the environment, okay? Ukiwa na jua kali sana, unapata in Kikuyu they call it kuhoha. That should be a better word, wilting. Okay, so the plants will start drooping. So you'll find that the plant leaves will start drooping. Why? Because they depend on water for support. Others, like plants, like trees, most of the trees, they have wood. So no matter how hot it is in the environment, you'll find that most of the plants will remain active without the leaves and the branches and the other parts of the plant drooping. But for herbaceous plant, or those plants that are made of don't have a wood, they will droop and they will wilt, okay? So therefore the cell wall makes the plant to have a definite shape, and also it provides mechanical support, and it also allows the plant to remain active since the plant cell will not burst. Good. So we have the nucleus. The nucleus, we said, is like the brain of a cell, because it is the one that controls what happens in a cell. So if a cell should stop allowing materials inside, or if it should start losing materials inside, if it should start carrying out for respiration, if it should continue carrying out photosynthesis, all those instructions will come from the nucleus. So the nucleus is responsible for controlling the activities of a cell. So if a cell has no nucleus, it will not survive. So why? Because the cell controls the function. That's why we have an NB here. So it is saying NB. A cell without a nucleus would only survive for a short time because it is not able to carry out normal cell functions. So from these, there's a common question that is usually tested in a SEMA. Explain what will happen if the nucleus of a cell is removed. So basically you'll say the cell would die. Now, let us look at the differences between plants and animal cells. So they are very simple because you're basically comparing and contrasting. Like for example, a plant cell is usually large, an animal cell is usually smaller. So we are not saying it is small, we are saying it is smaller compared to the plant cell. Then the other difference is a plant cell is regular in shape, while an animal cell is irregular in shape. Why will a plant cell be regular in shape? Because it has a cell wall that gives it a definite shape. While for animals, it is a bit irregular because it is made up of a cell membrane as the outermost wall. Then the other difference, a plant cell has a cell wall, while an animal cell lacks a cell wall. So a plant cell has a cell wall, while an animal cell lacks a cell wall. This is the outermost layer of a plant cell. And remember kindly that a plant cell is made of, a plant cell wall is made of cellulose. That is important. The other difference is that a plant cell has a large central vacuole, or what we called the subvacuole. Let me take you back with the diagram so that you can see. I'll take you back to the diagram where we have both the plant and an animal cell so that you can see what I'm talking about. So we've said a plant cell has a large vacuole at the center. a plant cell has a large vacuole at the center. Hapa hii yote. Okay, so this is the subvacuole. But when you look at an animal cell, it does not have a vacuole at the center. So a plant cell has a large vacuole as the center, and then an animal cell lacks a large vacuole at the center. That's another difference. Okay, now if you look at the two, you can see another difference. Another difference is a plant cell's nucleus is at the periphery. It is on the side, while an animal cell's nucleus is at the center. So a plant cell's nucleus is at the periphery, while an animal cell's nucleus is at the center. Okay, those are some of the differences. So we continue with the differences. The other difference is that plant cells have chloroplast, while animal cells lack chloroplast. Plant cells have chloroplast, while animal cells lack chloroplast. The reason why plant cells have chloroplast, we have said, is so that they can carry out photosynthesis, while animal cells lack chloroplast since they do not carry out photosynthesis. The other one, plant cells store starch, oils, and proteins. Underline the term starch. Plant cells stores starch, oils, and proteins. So I want you to underline these two terms in that statement. Underline the term starch and underline the term oils. Okay? Now remember at the beginning of the lesson I had said earlier that when plants carry out photosynthesis, they store carbohydrates in form of starch. Always remember that plants store carbohydrates in form of starch. So this means when a plant has carried out photosynthesis and formed carbohydrates, it will convert those carbohydrates into starch and then store them in form of starch. While as animals, we do not have the capacity to store starch. So instead, we convert our carbohydrates into glycogen. So both of them are carbohydrates, but they are in different forms. Reason being, they are being stored in different types of cells. Also, the one thing else you should also note is that plants store lipids in form of oils. That is why when you go to the supermarket, they'll tell you this is a Rina vegetable oil. This is a groundnut vegetable oil. This is a vegetable oil from maize. This is a vegetable oil from macadamia. Vegetable oil from whichever source. So if you get lipids from plants, they will be stored in form of oils. And then, whereas animals, we do not store lipids in form of oils. We store lipids in form of fats. I guess that's the reason why they say fat instead of oily. Okay? Yes, like I'm growing fat instead of saying I'm growing oily. You get? So animals store lipids in form of fats. Okay? So that's why we are saying plants store starch, oils, and proteins. When you talk about animals, it stores glycogen and fats. So instead of starch, plants store glycogen. Instead of oils, animals store fats. And then animals have no mechanism of storing proteins at all. At all. So plants will store proteins. As animals, we won't. So no matter how much proteins we take, then we will just excrete it. So I usually make this fun when I'm in class. I tell the students, when you go to a hotel or maybe at home during Christmas or a special event, and you eat half a kg of meat by yourself, only 77 grams of that meat will be used in your body because our bodies do not store amino acids. So nilikuwa nawaambia, if you take a lot of proteins like 1 kg of meat by yourself, the only thing you will enjoy is the sweetness from the meat. Otherwise, your body will only take little amount of the proteins, like 79 to maybe 100 grams, depending on how large your body is. And then the remaining 400 grams will go to waste. You will excrete them. So, and funny enough, Proteins are mostly excreted through urine. That explains the reason why when you eat a lot of meat, in the following morning when you urinate, your urine tends to have a very strong smell because it has a lot of urea. Sometimes it can even burn you when urinating because you did not take enough water. So therefore, from today now you know that bodies do not store proteins. So no matter how much you eat, it will only take what it requires, the rest will be excreted. This explains why some of us will have an issue with proteins. So when you eat eggs and you eat more than you need, you start having allergic reaction. So unaanza kuwa na pimples. This is because you have produced a lot of waste material, which is nitrogenous waste, which must be excreted, and some will have allergic reactions with the body. So therefore, what happens is when you take a lot of proteins, you will not store them in the body. You will lose them through excretion. So it is a bit advisable when taking proteins, just take a moderate amount, or you can just take it just for fun and the sweetness. Okay, yes, but our bodies will not store proteins, but for plants they will store proteins comfortably. Good. The last difference between plants and animals is that plants lack centrioles while animals have centrioles. Now, centrioles are found inside the nucleus and they are used in the process of cell division. So centrioles are cell organelles which are used for the process of cell division. So plant cells lack centrioles while animal cells have centrioles. So you can note that centrioles, so that when we'll be talking about cell division process, because we are going to see how our cells divide, we will mention centrioles a lot. So therefore, plant cells have no centrioles, but animal cells have centrioles. Those are just the differences. So with me here, I have study questions. I hope we can go through them together. So study the diagram below and answer the question. So the diagram below represents a nucleus. So this is the nucleus now, the cell organelle. Name the structures labeled E and F. So structure E is the nucleolus. So nucleolus are found inside the nucleus, and it is in the nucleolus that you are going to find your DNA. Now, something interesting about living organism is that we have DNA. We have what we call deoxyribonucleic acid. What is the importance of DNA? DNA is very important because it is the one that contains our makeup. Why is it that you are light skin? Why is it that you are dark skin? Why is it that your hair can grow very long? Why is it that you are able to, maybe for example, last a longer period of time in the field because you can play a lot and others can't even play a lot? So how is it that you're very good in something? Why is it that your eyes are brown? The iris of your eye is brown. Why is it that the iris of your partner or your friend or your children, why is it that they are gray? Others, like Europeans, you'll find that they are even hazel. Others are even blue. So it depends on the genetic makeup. What makes you up? Our genetic makeup is stored in structures known as chromosomes. And inside the chromosomes, we have DNA. Okay, so DNA is found inside the nucleus, and not in the nucleus, but inside the nucleolus. So the nucleus is the bigger one, and then inside the nucleus we have a nucleolus that contains our DNA, that controls how our cells are, our behavior, our characteristics like skin color, hair length, how tall we can grow, etc. So then we have structure F. Structure F, we've said it is the nuclear pole. What is the function of a nuclear pole? Any pole controls entry and what leaves. Okay, so inside the nucleus, some materials can pass through while others will not pass through. Like for example, when there's something we call cell division. We'll be learning about cell division. Because the DNA is found inside, the DNA is found inside the nucleolus, which are found inside the nucleus. There's something we call chromosomes or ribonucleic acid. Chromosomes have nucleic acids, which control how we are, how our cells are. That is why if you're dark skin, uko dark skin mwili yote, unless maybe you had a scar. So all those processes of how many cells will be formed, what will be the color of the cell, what characteristics will the cell have, those information will have to pass through the nuclear pole from the nucleolus. So we're going to talk about that in the later contents, okay? But just know that a nuclear pole allows entry of materials into the nucleus and also allows some materials to leave from the nucleus to the outside, now to the cytoplasm, okay? So we have the nuclear pole as F, and then we have the nucleolus as part E. We move on. So the other question is, state the function of part F. So part F is the nuclear pole. So the function is, it facilitates movement of materials in and out of the nucleus. So materials which are small enough will pass through. The ones which are so big will not pass through since they cannot fit. State two main functions of the vacuole, okay? So of course they are talking about the vacuoles which are found inside plants and animal cells. So tulisema that vacuole stores salt and sugar, okay? It stores salt and sugar. So that vacuole controls what gets inside. Like for example, in plant cells, it controls what gets inside the plant cell and what leaves the plant cell. Also, we have other types of vacuoles, like for example, food vacuole. Okay, like for example, we have some vacuoles which will store food. Sawa sawa. Then we have a very special vacuole that is found in animals that live in fresh water. A very good example ni amoeba. Okay, move with me. I've said there are very many types of vacuoles. We have vacuoles which store sugar and salt, like the contractile vacuole. These ones, they control what gets inside the cell and what leaves the cell in terms of concentration, like we are going to see when we'll be talking about cell physiology. Because when you separate two solutions, one which is highly concentrated with sugar and another one that is highly concentrated with water molecules, there will be movement of materials, like we will see. Like water will move from where it is highly concentrated to where it is lowly concentrated. Don't worry about that, I will explain that later. But just know there are some vacuoles which control movement of materials based on purely concentration of those two materials, okay? Then we have some vacuoles that store food, okay? Others digest food, sawa sawa. And then we have a very special type of vacuole known as the contractile vacuole. And this contractile vacuole will be found in plants that live in water, aquatic plants. For example, an example of a plant that lives in water, of an animal that lives in water that uses a contractile vacuole is an amoeba, okay? Amoeba. Let me see if I can show you something on amoeba. Let me show you, just a minute. I want to draw for you amoeba. I want to draw an amoeba for you and show you. Just a minute, I need to stop sharing so that I'll show you. Kindly be patient. Good. Good. Amoeba. So, amoeba is an aquatic organism. So, meaning it lives in water. Aquatic organism means it lives in water. So, we have that. So, this is fresh water. Okay, this is amoeba. Higher. Now, these animals, amoeba is an animal. It's an animal. It is unicellular. First of all, you should note it is unicellular, meaning it is made up of one cell. It is not multicellular as human beings. So what happens is, this animal, because it lives in fresh water, water will have to move inside, okay, because it is living in water. So at some point, the cell membrane will allow water to move inside. So this animal cell, it will gain water. When it gains water, it can burst because it is getting too much water. Remember, we've said animals do not have a cell wall. They instead have a cell membrane. And a cell membrane is not that strong. So therefore, it means if this animal continues to take in water, it might burst because it lacks a cell wall. However, it has some very special structures known as the contractile vacuoles. Okay, that's how a contractile vacuole will appear. So a contractile vacuole. Now the function of the contractile vacuole is ile maji yenye inaingia ndani ya amoeba, instead of making the amoeba to swell and burst, that water flows into the contractile vacuole. There are very many. It's not a single contractile vacuole. They are usually very many. So if we have another contractile vacuole there, it will also gain water or drain water from the cells, from the amoeba cytoplasm. So we'll have very many contractile vacuoles like that. Another contractile vacuole. Another one there. So very many. So what happens is, all these contractile vacuoles will drain the water from the amoeba. So amoeba inapata maji kutoka kwa environment, hayo maji yanaingia kwa contractile vacuole. When the contractile vacuole is full of water, it is the one that bursts. So when it bursts, it releases the water back to the environment. So water gets into the amoeba through the cell membrane. Then the contractile vacuoles inside the amoeba gain the water. They drain the water from the amoeba. They get filled with water and they burst to release the water to the environment. And that explains the reason why freshwater animals do not burst, especially the amoeba, despite the fact that they live in aquatic environment, because they gain water. When they gain water, that water will be excreted or removed by the contractile vacuole. Contractile vacuole. Sawa, contractile vacuole. So these ones are very important in aquatic plants, especially, not aquatic plants, aquatic animals, especially the amoeba or unicellular organisms. Okay, that's why we're saying the other function of a vacuole is excretion of excess water. Okay, excretion of excess water, which is removing the water which is gained by these unicellular organisms. Very good. So I hope you have understood about the contractile vacuole, and that is another function of the contractile vacuole. So let me go back to the document I was sharing so that you can continue doing those questions that were there before we move on to something else. So let's see. Very good. Good. Higher. So the other question was, name the plant cell organelle that stores chlorophyll. Name the plant cell organelle that stores chlorophyll. Now remember, we said chlorophyll is the green coloring matter in plants, and the importance of a plant having chlorophyll is so that it can trap sunlight for photosynthesis. Okay, so therefore it means the only plant cell that contains chlorophyll is the chloroplast, because chloroplasts are the ones we said are the sites for photosynthesis. So the plant cell organelle that stores chlorophyll is the chloroplast, and the importance of chlorophyll is to carry out photosynthesis by trapping sunlight. The other question is, name the plant cell organelle responsible for intracellular digestion. Intracellular digestion. Intra means internal. Intra means internal. This word intra here. Intra means internal. So internal, intracellular. So it means internal cell, responsible for internal cell digestion. And in this case, when you talk about digestion, we're talking about breaking down of excuse me, breaking down of the internal cell organelles. Okay, responsible for intracellular digestion, breaking down of internal cell organelles, and that cell organelle is lysosome. So we say that the lysosome contains lactic acid, which breaks down the cells or the cell organelles that are worn out. Okay, yes, so that is the end of those study questions, but I hope you have learned something from those study questions. Very good. So we can comfortably now move into something else. Preparation of temporary slides. Another area that is highly examinable. Now, what is a slide? Remember when we were talking about the microscope, tulisema that a microscope observes a specimen. And we do not place a specimen on the stage. Now you know the stage because we learned about the parts of a microscope. We do not place the specimen directly on the stage. We said there's usually a glass slide, and this glass slide is rectangular in shape like that, and we place the specimen at the center. And then this is what we take and place on the stage. Are we together? So this is what we call a slide, the rectangular glass that is used on a microscope. So we call it a glass slide. A glass slide. But for us to be able to observe what is it in a specimen, we mount it. We call it mounting. Placing a specimen on top of a slide is known as mounting. So when we are mounting, we are basically placing a specimen on a slide, that rectangular glass. Okay. Now there are two types of slides. Now when we use this slide, we can either use it using a permanent. We have a permanent slide, types of slides. We have permanent slides, or we have temporary slides. What is the difference between a permanent slide and a temporary slide? Temporary is something which you just prepare and you later discard. But for permanent, it is something which you will use for a long period of time. So this is what happens. So in the lab, if you want to observe a specimen just for one day, especially using fresh specimens, you just place it on a glass slide and observe it one time. That is a temporary slide. So this one is for immediate use during a laboratory exercise. So you take a specimen, place it on a glass slide, mount it on the stage. Mounting tumesema ni kuwekelea hapo kwa stage. So you mount it on the stage and then you observe what you're observing. Okay. Then there's a permanent slide. A permanent slide is one which has been prepared with the name of using it for a long time. So when you're preparing a permanent slide, you prepare it in such a manner that it will last for a long time before it can be thrown away. So mostly permanent slides are commercial. They are made in industries or in places where they are able to make these slides, and then they sell them to schools or to individuals so that they can be observing them. Now, one of the things you should note about these permanent slides is mostly they are used on plant sections, okay? But you cannot use permanent slides on animal sections. But sometimes you can have, for example, if it is a worm or a parasite, like a ringworm, so you can take those parasites. Of course, they should be dead because you're observing using when you're preparing them, you kill them and place them in slides which will distribute. Ndio mtu akitaka kuona venye ringworm inakaa, they can easily observe from the permanent slide. So bottom line here is a permanent slide lasts for a long period of time, can last for years, while a temporary slide is used temporarily just for a setup in the lab, and then you discard it. Okay, good. Now, what is the procedure carried out when preparing a temporary slide? So you've gone to the lab, you've been given kale, sukuma wiki, and you've been told to make a section of a kale so that you can observe the stem of a kale. So umeingia kwa lab, the specimen is kale. We're going to look at this when we'll be doing practical. So this is a very important procedure, because in a KCSE exam like last year, you can be told to prepare a slide. Last year they were told to prepare a slide, so they were given kale, they were supposed to section it, and then they were supposed to observe the stem of that kale. So what we should do here, in case you've been told to prepare a slide, you take the specimen, if it is the kale, and then you follow the procedure which they have told you or given you in that paper. However, there are some things which you need to know before you even step into a class where, or into a laboratory to prepare a temporary slide. So procedures. The first thing, we call it sectioning. Okay, sectioning is basically chopping or cutting. So cutting is what we call sectioning. So you see, when you're observing a specimen in the lab, you cannot observe a thick specimen. Like uchukue the whole of the kale and you place it on a slide, it will not be visible. Reason being, if you use very thick sections, then cuttings of the specimen, light will not pass through. So when you're cutting a section of a specimen to observe in the laboratory using a microscope, you must make it very thin as possible, so that light will pass through. We will do this practically, okay? Our very first practical that we're going to do, we will do it using, it will be about preparation of temporary slides, and we're going to use onions, okay? Yes, good. So you take a very thin section. So when you're sectioning or when you're cutting your specimen, you should make sure that it is as thin as possible. What is the importance of making thin sections? The importance is making thin sections allows light to pass through. Okay, it allows light to pass through. So in an exam, they have a funny way of testing this. If they don't want you to make a section in the lab, they ask you, what is the importance of carrying out the following procedure processes while preparing temporary slides? Wanakuandikia making thin sections. Your answer should be to allow light to pass through. Nimesema one of the way they can test is wanaweza kuandikia what is the importance of carrying out the following procedures ama making thin sections when preparing a temporary slide. So the answer should be to allow light to pass through. Very good. So when you're making a section, you should use a sharp razor blade to ensure the cells are not distorted or damaged. Okay? When you're sectioning or when you're cutting a specimen, two things should be done. Make sure you make it as thin as possible so that you will allow light to pass through. Also, make sure that you use a sharp razor blade to ensure the cells are not damaged or distorted. Very good. Step number 2, or something else you should ensure you have done when you're making a temporary slide, is make sure you place that section in water. So when you cut a specimen into a thin section, make sure you have placed it in water. Why do we place it in water? This is because the sections can lose water or become dehydrated. And when the section becomes dehydrated, cells will shrink and clump together, making it very hard for you to see the different parts of that specimen. So therefore, to ensure that the cells are not dehydrated, immediately you make a section or you cut a part of a specimen, you should place it in water. So in an exam, they can also test this content by asking you, what is the importance of placing sections in water? while you're preparing slides. You'll say to prevent dehydration of the cells of that specimen. Okay. Another thing that you're supposed to do when you're preparing temporary slides is staining. Okay, staining. Staining is dipping that section in dyes. Okay, you know dyes. Dyes are basically like colors. So usually we use iodine. I'm sure at some point either you or someone that you are close to has ever been injured and you've taken them to the hospital and the doctor has used a brown liquid to clean their wound, or even you have done that. That brown liquid is what we call iodine. So one of the most important and the most common stain or dye that is used in preparation of specimens is iodine. So what happens is when you make a very thin section, it is very transparent. Why? Because you have made it very thin. Because we want to see what is inside that specimen or that section, we paint it with this dye. Painting doesn't mean you take a brush and paint. No. Basically, you take a drop of iodine and you place it on the specimen. So that drop of iodine will color that specimen and it will make the internal parts of the specimen very, very clear. So therefore, staining is putting dyes on a specimen to make the internal parts of the specimen clear or distinct. So most commonly used stains are iodine solution, we have neutral red, and we have eosin. So in an exam, they can ask you to state the most common stains used in preparation of a slide. You can say iodine, neutral red, and eosin. So the reason why we stain specimens is so that we can make the internal parts or the different parts of that particular specimen to be distinct and clear. So the other thing that you do when you're preparing a temporary slide is mounting. Okay, mounting. We have said the first thing you do is use a very sharp razor to cut a section. Let's assume we are making a section of a kale. So you take the kale, unachukua the stem, okay, or the petiole of the kale, and then you cut a very thin section, kadogo kabisa. Why thin? To allow sunlight to pass through, ama to allow light to pass through when observing in a specimen. For you to be able to make a very thin section, you use a sharp razor blade so that you make a very thin section and you avoid damaging the cells. After you make that section, step number 2, you put it in water. Place it in water. Why do you place it in water? So that the cells do not dehydrate, to prevent dehydration of the cells. After you have placed it in the water and now you are ready to observe it, you take it out of the water, place it on the slide. Remember we've said the slide is a glass, rectangular glass, where we place the specimen. Okay, so place that section on the slide and add a dye. An example of a dye is iodine. Why do you add a dye or a drop of a dye on that specimen? To make it distinct and so that you can be able to observe the internal parts. From there, you cover that specimen with something we call a cover slip. You cover it with something called a cover slip. I want to draw for you a cover slip, so allow me to stop the broadcast or the share, sorry, and then I will show you a slide. I should show you the slide. Sorry for that. Good. So we have a rectangular glass, which we've said this is a glass slide. Okay. When you take your specimen, you put it, like I was explaining here. So if this is your kale, when you're preparing a slide, you cut a very thin section, kadogo kabisa. You cut a very thin section, that is sectioning. You take that thin section and dip it in water. After you have made the section and now you're ready to observe, you place it here on the slide. Okay, like that. When you place it on the slide, you add a dye, a drop of a dye on this specimen. So unaweka dye hapa so that you can make that specimen clear. After you have placed the dye there, now the specimen is ready for observation. But before you place it on the stage of a microscope, you need to cover it with a small glass known as the cover slip. So this is the cover slip. Okay, that is the cover slip. So a cover slip's function, kazi ya cover slip, its work is to, number one, make sure that there is no dust that is coming into contact with the specimen. Because if dust comes into contact with the specimen, unaweza pata you are observing four parts of the specimen. Ni dust, lakini unaona ni kama ni internal structure of that specimen. So therefore we place a cover slip so that the specimen does not get any contamination or any dust. Sawa sawa? Very good. And then now you take the slide and you place it on the stage of a microscope for observation. So tumesema procedure is you make a thin section, a very thin section to allow light to pass through. You place that section in water to prevent dehydration. After you place it in water and you're ready to observe it, you place it on the slide and you add a dye. A dye can be eosine, it can be iodine to make it clear. And then you place a cover slip to protect the specimen from dust and other contaminants. Okay, and then you take the glass slide and you place it on the stage. Placing the glass slide on the stage is known as mounting. So mounting is placing the glass slide on the stage. So those are the procedures that you follow when you are preparing a temporary slide. So I hope today we have learned a lot of things, because I'm sure we have grasped based on what we've been doing previously. So today we have learned about some cell organelles. Okay, we've also gone ahead and talked about functions of those cell organelles. So we've also gone ahead and talked about differences between plants and animal cells. And finally, the last thing we have done today is preparation of temporary slides, where we have said a temporary slide is a slide that is used to observe a specimen and then discarded. And then we said that we have another one known as the permanent slide, which is preserved for later use. So we also went ahead and talked about the procedures to follow while preparing a temporary slide and the importances of those procedures, like sectioning, making thin sections, and we do that to allow light to pass through. Also adding dyes so that we can stain the specimen, adding water to prevent dehydration, and mounting, which is placing the specimen on a slide and then placing it on the microscope. Okay, good. We have this one here, advantages of using a cover slip, so that you can hold the specimen into place and you protect the specimen from dehydration and contamination with dust particles. Okay, now when we meet during the next, now look at this before you leave. There's a practical activity aim to prepare and observe temporary slide of onion cells under the light microscope. So when we prepare, when we decide that we are going to have a practical activity, the first practical activity that we are going to do, so that we can learn how to use a microscope and at the same time learn how to prepare a temporary slide, will be this practical activity. So this one is a must we do, so that we can learn how to handle a microscope, and also we will learn how we prepare temporary slides. So that is the practical. We will talk about that. So when we meet during the next lesson, we will talk about so these questions here on estimation of cell sizes, I prefer we do it after we have learned about how to prepare a temporary slide because they are connected. So we are going to skip that bit. We will do it when we'll be doing the practical. So after this, when we meet during the next lesson, we'll talk about cell specialization. I mean, how do we, why do we have some cells having specific characteristics? Why do we have some cells appearing the way they appear? Why is it that some cells can be able to perform some specific functions and others can't? Cell specialization is basically why cells appear the way they are and what functions they perform. Why are they made in a particular physical form? So when we meet, we'll talk about cell specialization and we'll look at examples of specialized cells in plants and animals and see their importances. So with those very many remarks, because I know some of us are leaving for work, I want to end the lesson there. Thank you for showing up. And if you have any question, this is the chance to ask the question. And if there isn't, I want to wish you a good day and may you be protected and guided throughout. Thank you. Well, looks like there's no question. Have yourselves a beautiful day and take care of yourselves. Bye.